Thanks very much, Claudia, and thanks also to the organising committee for inviting me. Just some disclosures. I have funding from various government organisations and agencies, charities, and also relevant to this meeting, I chair the Population Systems Medicine Board at the Medical Research Council of the UK, which deals with nutritional matters. So what I'd like to do this morning is give a little bit of context as to why one might be interested in the exposome, tell you what the exposome is, and focus particularly on the metabolomics, what I call the nutriome, that looking at nutrients in relation to outcomes, and a little bit about why we might be also interested in the genome, as that gives us an anchor point to look for causal pathways, and then finish off talking about some big cohorts that are being established and that provide a vehicle for carrying out this type of work, and then a few key takeaways at the end. So what is the context? So we know that in terms of chronic disease, there have been very large fluctuations. So there was the, if you like, emergency of the epidemic of cardiovascular disease in the 50s and 60s, which became very apparent in North America, but also in Europe. And since then, from the 1950s and 60s onwards, there's been a remarkable decline in cardiovascular disease in many countries. So US is the orange one, something like a 2/3 decline in instance of cardiovascular disease. But that has been shared in many other countries, but not always at the same time. So for example, there was a continuing increase, this is Hungary, in countries of Eastern Europe, and only now has the decline started. So if you look across Europe, there is remarkable differences in the rates of heart disease from Eastern Europe through to Southern Europe, which is characterized by the Mediterranean diet. And there's something like a 6-fold difference in mortality and incidence rates. And these are genetically very similar populations. At the same time, we've seen this massive increase in rates of overweight and obesity. Something like a 3- to 4-fold increase in rates of obesity across the world from 1980s through to the present day. So clearly this is going in the wrong direction. And more generally, if we look at across the world, we see this transition, this so-called epidemiologic transition from infectious disease and high mortality in infancy and around childbirth, people surviving through to young adulthood and then into older adulthood, and then they become susceptible to chronic diseases. So there's this massive switch from the infectious disease to the chronic disease, a so-called grand challenge in non-communicable disease. And here you see, obviously, smoking and overweight as one of the major issues that we have to address, not only in developed countries, but also in the developing world. And this slide summarizes the upstream and proximal risk factors for cardiovascular disease, because clearly we see these diseases happening within the context of society. Things like living conditions, the natural and physical environment, including air pollution and noise pollution, which is a current focus of research, temperatures with climate change and increasing temperatures across the world, affecting incidence of cardiovascular disease, and of course the social environment, because these diseases cluster amongst poorer people. And then if we look at the more proximal behavioral factors, the things that we know about, smoking, alcohol, physical inactivity, but also poor diet that you've been discussing earlier today and we'll be discussing through the meeting, adiposity, and then we get to the more proximal physiological pathways that we can measure, blood pressure, lipids, blood glucose and diabetes, and inflammation, leading to subclinical atherosclerosis and ultimately to clinical disease. So we need, if we want to maintain the improvements in cardiovascular disease and also prevent a resurgence of an epidemic of cardiovascular disease, clearly it's still the number one major cause of death around the world, then we need to address the upstream determinants, but also the proximal determinants. And there was discussion before the break about biomarkers to develop appropriate biomarkers so that we can monitor the progression through subclinical atherosclerosis before clinical disease is onset. And if we think at the genetic level, we know there is genetic susceptibility to these diseases, to chronic disease in general, to cancer, to cardiovascular disease, and through the onset of the genetic genome-wide association study, we know there are hundreds of genetic markers of these diseases, each with a very small, mainly for the chronic diseases, a very small effect, but nonetheless a detectable effect. And if you look at the various diseases, they seem to cluster genetically, and there seems to be a common genetic markers across cardiovascular, metabolic disease, and in some extent cancer. But against that genetic susceptibility, we know that it is the environmental factors which are really important in determining who gets disease, who does not get disease, and both at the population level and at the individual level. And this was summarized by a diabetic doctor, famous diabetic doctor from the US, Elliot Joslin, way back. Genetics loads the gun, that's the genetic susceptibility, but environment pulls the trigger. So if you live in a place, for example, if you live in the Amazon basin and you're Yanomamo Indian, your blood pressure does not rise with age. But if people move, so for example, the Kenyan Luo moving from their villages, rural villages to Nairobi, in their rural villages, their blood pressure is protected, their blood pressure does not rise with age. But as soon as they move, very quickly their blood pressure goes up. So we know that they may have genetic susceptibility, they may have genetic protection, but as soon as they hit an environment which is conducive to chronic disease, then very quickly the environmental factors working against the background of genetic susceptibility makes one susceptible to these diseases. So we need to address environmental factors underlying the high prevalence of risk factors and high morbidity mortality from these diseases at older ages. So we have a major challenge because actually the genetic data, though there's a lot of it, so it's a big data problem, are pretty simple. They're a 0, 1, 2 fixed digital readout. But the environment changes over time. It's very difficult to measure. And as we take the measurements, you have a dynamic range and it's continuously distributed and it's occurring over the life course. So we have very imprecise and poor measures of these environmental factors, including dietary factors, which are notoriously difficult to determine and collect through standard measurement techniques. So we do need new methods and approaches to capture the effects of environmental exposures on non-communicable disease. So the concept is that we have these lifestyle and lifelong exposures related to where we live, the sort of culture we live in, but also our individual preferences. And that has effect at the molecular level. That will then perturb metabolic pathways, which we can maybe capture at the systems level and maybe identify biomarkers of exposure and also early biomarkers of effect, disease biomarkers. And that determines our molecular phenotype, which, as I say, is on the trajectory from health to disease. And ultimately, that will determine the clinical phenotype, whether we progress to actual disease, clinical disease. And we need to work across all these systems to understand this relationship, and potentially and hopefully then we can intervene in terms of policies, improved policies to protect health, and ultimately improved health at the individual level, because this could be personalized, and at the group population level. So moving on, I'm going to focus on the metabolome. And what is the metabolome? So metabolomics is the capture and measurement of small molecules in biological samples, most commonly in blood, also in urine, but it can be in other samples. And the metabolites represent the downstream end products, biochemical end products of metabolism across all systems, if you're capturing it in something like blood or urine. But it's the closest to the phenotype in terms of these omics. So you start here with the genome, the epigenome, which talks about how we transcribe the genetic information, the transcriptome, which captures that transcription, the proteome, the proteins driven from the genome, and then downstream metabolism is captured through the metabolome, which will be affected by things such as your age, your sex, your ethnicity, your diet, physical activity, stress, in utero effects, because this is over the whole life course, external xenobiotics such as pollution and drugs, where you live, and also the gut microflora, the microbiota. And what it does, it gives the link between these environmental stressors, your intrinsic metabolism, your genetic information, and that transfer between the healthy state and the disease state. And conceptually, you can think of it as you have ideal health through your life course, might be represented by this line. No one ever achieves ideal health. So healthy people might be working along this trajectory over the life course, but unhealthy people might be going on a much worse trajectory. And what we want to be able to do is to capture these people early on in their life course and understand who is going down this route versus that route, potentially intervene, and hopefully then recover a healthy profile, which will lead to prolongation of healthy living and reduction in early disease, cardiovascular, cancer, or whatever. So how do we carry out the metabolic profiling or the metabolomics? So the two main techniques are through nuclear magnetic resonance spectroscopy and mass spectrometry. They capture different things, they have different sensitivities, but the idea is that you have a stressor, or in an experimental situation, you might be looking at a knockout animal model. You might be diseased, which will affect the metabolome, or there might be other sources of toxicity. That has an effect at the cellular level and the tissue level, and ultimately that will spill out into the biofluid affected by external stressors such as diet, the gut microbiome, other chemicals, and then we capture the metabolic profile, as I say, at one point in time, or preferably we capture it at multiple points in time during the life course. We then get a spectral readout from NMR, which is one-dimensional, and from mass spec, which is two-dimensional. But the challenge is we capture, if you're doing so-called untargeted metabolomics, where you're capturing the whole spectrum, you're getting huge amounts of information. It's very information-rich, but it's complex. And the challenge is to retrieve useful data from these signals. So for NMR, you might capture thousands of signals representing hundreds of metabolites. And for mass spectrometry, maybe in the tens of thousands of signals and maybe the thousands of metabolites at various levels of sensitivity. So I mentioned you can do this metabolomics either in targeted form, where you test for specific metabolites and pathways, which is clearly attractive, but misses a lot of the information that is out there that you potentially can capture. Or you go through untargeted mode, where you capture everything, but then you have the problem of finding signals and trying to interpret those signals. So we have the analytics, which is via NMR or mass spectrometry. We will discover new biomarkers against an outcome of interest, for example, cardiovascular disease. And then we need to identify those biomarkers. And this can be quite a complex process because you've got to do structural elucidation of those signals. You have to characterize those biomarkers. As with any biomarker discovery program, you need to validate those biomarkers in independent cohorts and then potentially test those biomarkers as potential predictive markers of future disease. So the steps in the analysis are to use these technologies, and in my declarations I'm a co-investigator of what's called the MRC NIHR National Phenome Centre in the UK, where we are working with industry in terms of the instrument manufacturers, Waters for mass spectrometry, and Bruker for NMR, and using state-of-the-art methods to capture the information from biological fluids, as I say, mainly blood and urine, to get so-called fingerprinting of those fluids. And then you combine those data with extensive personal characterized data, mainly from cohorts, large cohorts, on clinical information, lifestyle information, but also other omic data sets. So this is a big data fusion problem. And then carry out bioinformatic and pathway analyses to place those metabolites into potential causative pathways. And clearly this is a complex program. The methods are in some ways bespoke, and so it's extremely important that these methods are standardized across both the NMR and the mass spectrometry, and across different laboratories who are doing this sort of work. So we're setting up what's called an International Phenome Centre network to develop these common methods and to develop these common standards so that we can get an international network that can do this sort of work. And here's an example from the Intermap study, which is a study of diet and blood pressure in 4 countries, the UK, the US from the west, China and Japan in the east, 17 population samples involving over 4,500 men and women in the age range 40 to 59. And we collected multiple blood pressure measurements. We collected multiple 24-hour dietary recalls by face-to-face interview. And we also collected 24-hour urine collections, some on 2 occasions, 3 to 6 weeks apart. And I'm going to focus on the urine collections because those we've analyzed using NMR spectroscopy for the metabolome. But just to mention in passing the results for the 4 countries from the 24-hour recalls. So this is China, Japan, the UK and the US. The purple bars are the % calories from sugar. This is the US, this is China. Clearly very different diet, as you know. This is the starch intake, % calories in China, and this is in the US. And down here we have the vegetable intake much higher in China than in the US. So very, very different diets, as you're all very well aware. And this is taking the total urinary metabolome for each of these 17 population centers and doing a hierarchical clustering analysis to see how they fall. And you can see that the eastern samples were differentiated from the western samples. Sadly for the UK, we ended up looking just like an American center. So we became from sort of our metabolome, we are the 51st state. Here's the UK. And you can see that, so in the US, there's some clustering. These are females, these are males. There's some clustering by gender. And here in Japan, again, you've got some clustering by gender, and in China, the clustering is only by place. Now, interestingly, the Chinese and the Japanese, who are genetically quite similar, are distinct in terms of the metabolome, and the Chinese distinct from the West. But this group here, the Honolulu sample from the US, are Japanese Americans, and they map like Americans. So the metabolome is reflecting their lifestyle, mainly not their genetics. Though interestingly, they're the closest mapping of the Americans to the Japanese. So there's some genetic signal that's appearing in the urinary metabolome, but it's mainly a lifestyle signal reflecting the fact that they're living in a US environment. And in China, the north and the south of China were distinct despite being genetically very similar. And if we look at that further on this plot, PCA plot, you can see that the north of China is very much distinguished from the south of China. And the north of China, they have higher risk of cardiovascular disease and stroke, they have higher blood pressure, they have much higher urinary sodium excretion, much higher sodium-potassium ratio, lower calcium, lower magnesium excretion, and as I say, higher risk of stroke and cardiovascular disease. And metabolically, they're very different, but genetically, they're very similar. So within that study, we then looked at the signals which were differentiating the different populations and then related those signals to blood pressure. And for example, we found this association between urinary formate excretion and blood pressure, and this had not been identified previously. So this is, if you like, discovery, and using what we call the metabolome-wide association approach, using agnostic approach to look at all the possible signals in relation to the outcome, in this case blood pressure, and then adjusting for the fact that we'd done this multiple testing, and came up with a hypothesis that formate was involved in blood pressure regulation, and it so happens that formate is involved in the chloride exchange in the kidney, so there's a potential mechanism. And looking now at measures of adiposity and obesity, so this is looking at body mass index as a marker of adiposity in the same population. This is in the US Intermap sample. Again, looking at the urinary metabolome, not looking at obese versus non-obese, but looking across the entire range of body mass index from low to high. And with BMI as a marker, you get this very, very intense signals. So the red signals here, this is the NMR spectrum. The red signals are higher with higher BMI, and the blue signals are lower with BMI. Right across the spectrum, you find these signals, and if we put those into some sort of pathway analysis, here you can see, so the signals that we detected are either red for higher or blue for lower, and every system is disturbed as your BMI goes up. So this is involving muscle metabolism, mitochondrial metabolism, lipid metabolism, the Krebs cycle, etc. And interestingly, there's also gut microbial co-signatures which are also involved in this. So this is a stress. This is not just looking at obesity. This is right across the range of adiposity. There are multiple systems involved. Most of these are probably secondary rather than primary, so they're not causal. They're secondary to increasing BMI, has system-wide metabolic and molecular disturbances associated with it. And I mentioned at the beginning that we also, on many of these cohorts and individuals, have genetic data, mainly from so-called GWAS, genome-wide association chips. And we've been very interested in using those genetic data to try and inform causal pathways in relation to how those genetic signals, which we know are related, for example, to coronary heart disease, which is this group over here, or this signal here is related to Alzheimer's disease, can we get information from the metabolome that will help define these pathways? Because many of these genetic signals, we know nothing about how they are involved and how they increase or cause disease. We know they are causal because unlike everything else that we measure, the gene is fixed. So the gene is fixed and they relate reliably and in a cross-validated way to outcome, then we know that we're on a causal pathway. But we don't know what the intermediate effects are. And if we can outline those pathways, maybe we can intervene either with new therapies or with preventive approaches and lifestyle approaches. So this is looking at a targeted analysis based on so-called Helmholtz metabolomics database at various single nucleotide polymorphisms that have been detected in relation to coronary artery disease, and various metabolites have been identified, are associated with these SNPs, and there are some unidentified metabolites which would need to be identified to try and work out those pathways. And here's another example from our so-called airwave study, which is a study of 50,000 police officers and staff in the UK, looking at a SNP in a gene, ABCA7, which is associated with Alzheimer's disease, but about which we know nothing about function. And you can see, using one of the mass spectrometry analysis that we did, that we get very, very strong associations with some of these metabolites, all of which currently are unknown, but we're currently working out what these are. So this might give some new insight into the pathways linking this gene to the outcome. And just very briefly want to touch on the nutriome. And what do I mean by the nutriome? Well, again, this is the Intermap data. And what we've done is taken all the individual nutrients detected on the 24-hour dietary recalls and related these to blood pressure in an agnostic way. So just taken a whole bunch of the nutrients and related them to blood pressure and then adjusted appropriately for the multiple testing. And many of the things we find, we already knew about. For example, urinary sodium is positively associated with blood pressure, or in this case, the sodium-potassium ratio. And at this end, we have things like alcohol and iron intake and vegetable protein intake, which we'd already reported. But over here, we had a whole bunch of new signals that we hadn't thought about, hadn't detected previously, related to B vitamin and blood pressure. So again, using this agnostic discovery approach, you can find out new things, and we then went on to validate and replicate these findings in the NHANES database. And you can do other things with such data. So here's all the nutrients, and we've related that to the metabolome. And red means that they are positively associated and significantly with what comes out in the urine, and blue means they're inversely associated. So already we can then start looking at pathways between dietary intake along the bottom and the metabolites that we detect either in urine or blood. And over here, these are the amino acids, so what comes in comes out, but there are big white areas where what comes in doesn't come out in that form, and blue areas where what goes in has some metabolic transformation or use and comes out in a different form or is associated in a different way with the metabolites. So this is potentially giving huge amount of information about dietary intake and metabolic consequences of dietary intake in an agnostic way. And so this slide summarizes a couple of studies trying to detect new biomarkers of dietary intake, because, as I mentioned earlier, and as you all know, the physical measures of attaining information at the individual level of dietary intake are quite unreliable, depending on interview or recall or filling out questionnaires, and they're prone to bias and they're prone to inaccuracy. So it would be fantastic if we could develop new biomarkers of intake that could then be used to monitor people, as was discussed by Dr. Williams before the break. So here's one example where there was a trial, so this is all done in trial, randomized trial mode, where there was a standard diet, and then a standard diet plus fruit was tested, and a signal appeared in the urine, a metabolomic signature appeared, which turned out to be proline betaine, and that was then tested, and indeed proline betaine is contained in citrus fruit, and proline betaine turns out to be a very good marker of citrus fruit intake, of healthful diet. And here's another example published in the Lancet Diabetes Endocrinology last year, looking at, in a randomized trial, 4 diets. Diet 1 was like the DASH type diet, a healthy diet, and diet 4 was an unhealthy, usual American type diet. And again, looking at the urinary metabolome using nuclear magnetic resonance spectroscopy, and there were some metabolites which were higher in the healthy diet, some higher in the unhealthy diet. And then if you look at some of these, for example, hippurate, which is a marker of fruit intake, and it's a gut microbial co-metabolite, is higher in the healthy diet, and carnitine, on the other hand, which is a marker of meat intake, higher in the unhealthy diet. And over here you've got tartrate, which is a biomarker for grape intake. Again, that was higher in the healthy diet. So potentially at the sort of whole diet level, you can develop a metabolomic signature. This was then tested in 2 population cohorts and found to reflect DASH score in those cohorts. So potentially you could think of this as developing a ongoing biomarker of diet adherence to something like a DASH type diet or a healthy diet to follow people up over time. What happens to their urinary metabolome? Okay, so I just want to finish talking about cohorts, because this offers the substrate with which to test many of these possibilities in terms of these omic technologies, and to look at the exposome in relation to long-term health outcomes. So there are many initiatives around the world now looking at setting up and establishing very large cohorts to do this type of work. So in the US, you've got the Million Vets program, and you've got the Precision Medicine program, both looking at 1 million people. In the UK, we have so-called UK Biobank, which I'll come back to. Airwave study I mentioned to you. There's the EPIC study, another 500,000 study, which is across Europe. Kaduri study in China, another 500,000 study. Biobank Japan, 200,000, and the Korean Biobank, 500,000. So these are very, very large programs looking at following people up over time with biological samples, so that you can go back and look at the genome and the metabolome and the transcriptome and the proteome as potential biomarkers for future events as they accrue into the cohorts. So I'm just going to focus a little bit on UK Biobank. So Biobank collected clinical data at a clinical assessment centre for 500,000 people who were approached via the National Health Service. A lot of lifestyle information was collected, cognitive testing was collected, accelerometry was collected on 100,000. So it was really deep phenotyping. And of course, biological samples were taken, blood and urine, aliquoted and stored in, actually in a very large robot, which you can see here. And 100,000 of those 500,000 people are being invited back now to have MRI scans, DEXA scans for bone density and also muscle, carotid scans for early signs of atherosclerosis, and deep phenotyping of the retina, as you can see here. So very, very deep phenotyping with biological samples to follow those people up. How are they being followed up? It's being done mainly through record linkage. So as I say, we have the advantage of the National Health Service in the UK, so that we can go to cancer registries, we can go to pathology records, records of drugs, go to GP records, but also link into the cohort information about the xenobiotics that I mentioned, like environmental data, information about the built environment. So very, very rich information that will allow us to explore long-term effects of the environmental lifestyle exposures on health. We're currently looking, so at the moment, all 500,000 people have had a genome-wide scan using Affymetric chip, that we're now looking at doing full sequencing on those people, and also discussing now the metabolome as the next sort of study-wide test that we might try and do on UK Biobank. Now, as I said, as you can see, this is a big data problem, and to bring all this together needs a multidisciplinary approach. We need new methodologies. We're bringing together the computer people with the data people, with the health people. And the idea is that the whole is greater than the parts. But this is a massive effort and requires a whole new way of thinking and working if we're going to make advances, much as was discussed before the break. So my last slide, we do have these new technologies and methodologies using this omic data, which opens up fantastic opportunities for discovery of new biomarkers in relation to disease outcomes and intermediate outcomes for the non-communicable diseases. We need to apply these to these cohorts where we have embedded long-term follow-up, so we can look at the results from exposures many years ago and future health outcomes. And by taking advantage of all those cohorts around the world, we have the opportunity to validate and replicate, and that's extremely important, as we've learned from the GWAS studies. This will give us new insights on pathways and mechanisms linking environmental exposures to disease, the so-called exposome, and hopefully by getting that new information, we will be able to intervene better and earlier to prevent disease. But to, as I said in the previous slide, to take advantage of all this data, we need to have completely new ways of working across disciplines. So we need the physical sciences to work with the life sciences and the medical sciences to really make advances in this area. Thank you.
Thank you, Paul. Very, very interesting. We're going to take one or two questions. We're running a little short on time. If you could just introduce yourself.
My name's Gary Beecham. I'm from the Monell Center, and I really enjoyed your talk. I have a specific question, though, for you. I've had a long interest in salt, and obviously you have too. Is there any biomarker for salt intake? Just on a practical level, people really don't know how much salt they're consuming, and doing 24-hour urines is, for the average person, just not practical, and it's maybe not even true. Is there some progress in this area where you could have something that would be a marker that an individual or could use that would really reflect the sodium intake over time?
Well, as you say, I've been working on this for a long time. So we do believe that 24-hour urine is a good biomarker, but as you say, it's difficult to collect in population studies, and really you need, as you know, multiple collections, and that is difficult to achieve. We are looking at the spot urine in UK Biobank. We have 500,000 people, and as you probably also know, we've been looking at spot urine in relation to 24-hour urine because it is a much more tractable marker. Probably at the population level, it probably works. At the individual level, it's tricky. So, funnily enough, we do have an NIH grant to look at this question, to look at the urinary metabolome in relation to sodium intake. So I'm hoping that we will come up with a better biomarker.
John Erdman, University of Illinois. Actually, my question relates directly to population level versus individual level on metabolome. There's great value from the metabolome. But at the individual level, if I come and have my urine done and it was a stressful day, or I have a mild inflammation of some sort, I'm going to have a totally different readout. And just wondering if you could comment on the value at the individual level.
Yeah. So you're right. I mean, it only doesn't vary day to day. It probably varies, minute to minute or hour to hour. But actually, we looked at this within the Intermap data because we had repeated 24-hour urines, and to our surprise, it was remarkably stable. So the signals that we got looking at the first of those urines was very, very similar to the signal we got on the second of those urines. And actually, when we compared the two urine samples at the individual level, they clustered in metabolic space, similarly. So actually, I think there are minute-to-minute, day-to-day perturbations, but the underlying signal related to who you are and what you eat and what you do is, I think, quite constant. So that's very helpful for this sort of work.
Cho-san Koo with ILSI North America. Has new approaches in omics give us better indication of identifying salt-sensitive individuals?
Okay, so we haven't looked at that yet. My view on salt sensitivity is that you've got a whole range of responses to sodium right across the range. And it's artificial, in my view, to separate people into salt-sensitive individuals and non-salt-sensitive individuals. I think we all have a degree of salt sensitivity, if you like, insofar as if you do randomized trials of reducing sodium intake, on average, blood pressure will be reduced across the board. And I don't think it's that helpful to talk about salt sensitivity. It's a bit like looking at hypertension and non-hypertension. We know that the blood pressure outcome relationship goes right across the whole range of blood pressure from low to high. And it's the same, for example, looking at those adiposity biomarkers. It wasn't just if you're obese, you have this very different metabolome. It's right across the range of BMI. So my view is that, yes, some people will have a bigger fall in blood pressure, if they go on a lower sodium diet than others, but everyone ultimately will benefit. So my view is it's not particularly helpful.
Take one more question here.
Al Place at IMET. What about multiple associated biomarkers? In other words, not just one changing or correlated, but in fact there are multiple correlations among themselves.
Yeah. So that's the beauty of the taking that metabolomic untargeted approach. You get the whole spectrum, and obviously they're all highly correlated. So you've got a very correlated set of data, which is what we tried to get at with that network is that they're not working one at a time, obviously, as you say, but you're looking at a whole network perturbation. But you can capture all that if you look at the entire spectrum. But it's not easy. Working out what's really going on is not easy. It's all multidimensional. It's not unidimensional. But that's one of the big data challenges, I think, from this type of work. But you capture all the data, you capture all the information. Therefore, you can potentially make the right inferences.
All right. Thanks again, Paul.
Thanks very much.